Now, in this part of the video, I will be going to discuss the secondary growth. Now, this is a single topic which actually requires individual assistance, as it is a very tough topic and it is very hard to understand. So that is why I have divided this chapter into different parts so that you guys can easily understand it into simple and smaller steps. So the growth which basically helps in the which is done by the apical meristem and which or the primary meristem which helps in the increase in length of the plant organs is called the primary growth. Whereas the increase in girth or thickness of plant is called the secondary growth, and it takes place by the activity of cambium. So the tissue which is involved in secondary growth are two lateral meristem, which I told you that it is lateral meristem is a type of secondary meristem. These are vascular cambium and the cord cambium. So now, coming to the secondary growth in dicot stem. As you know that the dicot stem contains cambium, so the secondary growth is a typical feature of a dicot stem. And as monocot stem does not contain cambium, so the secondary growth does not take place in case of monocot stem. Now the secondary growth in case of dicot stem can be divided into two types. Secondary growth in the stellar region, that is in the stele part, that is below the endodermis, and the secondary growth in the extrastellar region, that is the region above the endodermis, that is in the cortex, hypodermis and all. So these are the two parts. Now coming to the first one, that is the secondary growth in the stellar region. Now a cambium which is present inside the vascular bundle is called the intravascular cambium. So as I told you that the cambium, this is the cross section, and you can see that this is the phloem and this is the xylem, and the cambium in between the phloem and xylem, this is called the intravascular cambium because it is present in between one xylem and phloem, and it is a type of primary meristem as it is formed from procambium. Now first of all, cells of the medullary ray becomes meristematic to form interfascicular cambium. Now the interfascicular cambium is the cambium which is present in between two vascular bundles. This is the interfascicular cambium. So this interfascicular cambium is a type of secondary meristem. Now this interfascicular cambium and this intrafascicular cambium together forms a ring-like structure, and this is called, and they together constitute the vascular cambium. Now the types of the cells are found in the ring of vascular cambium. Now basically two types of cells are found in this ring of vascular cambium. One is the fusiform initial and another one is the ray initial. Fusiform initials are long and pointed cells, whereas the ray initials is a oval shaped cell. Now continuous percranial division takes place in the fusiform initial. Now some cells are formed toward periphery called secondary phloem, and the some cells are formed toward the central axis and is called the secondary phloem xylem. Now by the activity of secondary phloem, primary phloem is pushed toward the outside and gets crushed. Now by the pressure of secondary xylem, all the primary tissue such as the primary xylem, pith, etc. degenerates in the center of stem. Due to this, the central part of the stem becomes woody, and this activity goes on continuously in plant throughout life. You can see that this was the normal cambium. The cells of the cambium divide to form the secondary xylem and the secondary phloem. Again the secondary xylem, again the cambium divide to form another layer of secondary xylem. So this results in pushing the or crushing the primary part of the cell, that is the primary xylem, pith, etc. And the growth of the phloem results in pushing the primary phloem towards the outer part of the cells. So this results in a growth in thickness, and this continues throughout the life cycle. Now secondary xylem forms in the plant regularly and primary tissues degenerates continuously. The new secondary xylem also degenerates the old secondary xylem. Now waste materials are formed in the stem such as lignin, suberin, tannin, etc. due to degeneration of cells. Because of this, wood in the central region of the stem becomes dark color due to the deposition of those secondary tissues, the secondary xylem, and also the lignin, suberin, tannins, etc. which are formed in the, due to the degradation of those cells. Now that wood is called the heartwood or duramen. Whereas the outer wood, the surrounding the heartwood is called the sapwood or the alburnum. Now heartwood provides maximum mechanical strength to the stem, whereas the function of sapwood is water conduction. And this too is very important. The difference between these two wood, that is the heartwood and the sapwood, is very important, and this is a very important question for your NEET exam. Now the cavities of tracheids and vessels of heartwood are progressively filled by waste materials. The bladder-like ingrowth of parenchyma cells enter the lumen of vessels and tracheids. Such bladders-like ingrowths are called tyloses. These are the tyloses, which actually blocks conduction through this xylem vessels in case of heartwood. That is why the heartwoods are unable to conduct any water and mineral, and the conduction is only done by the sapwood due to the formation of this tylose. Now, wood wood can be of different types on the basis of amount of parenchyma, on the basis of presence or absence of vessels, or on the basis of presence or absence of vessels. Now, on the basis of amount of parenchyma, the wood can be of two types: the monoxylic wood and the pycnoxylic wood. The monoxylic wood contains more living parenchyma. Sorry. The monoxylic wood contains more living parenchyma, and this wood are called softwood. For example, in case of cycas, whereas the pycnoxylic wood contains less living parenchyma, and that is why the wood is called the hardwood. Example, the commercially available woods like the woods of babool, the mango, etc. On the basis of presence or absence of vessels, the wood can be non-porous wood in which the vessels are absent. Example, the woods of gymnosperms. Whereas the wood in which the vessels are present are called the porous wood. Now the porous wood can be of two types: ring porous and diffuse porous. Now in case of ring porous, the vessels are arranged in a ring, whereas in case of diffuse porous wood, the distribution of vessel is asymmetrical, as in case of Azadirachta, that is neem. Now perclinal division also occurs in the ray initials. Now some cells are formed toward inner side and some toward the outer side to form secondary ray or vascular ray. All cells are parenchymatous and they help in radial conduction. So basically, by the activity of fusiform initial, secondary xylem and phloems are formed, whereas by the activity of ray initial, the secondary medullary ray or the vascular ray is formed. So remember this thing. Uh, Now the activity of cambium does not remain same. It is changeable all throughout the year. In winter or in autumn season, the activity of cambium is less, and the secondary xylem formed is not extensive through the vascular cambium. So this type of wood, the wood formed during this season, is called the autumn wood or the late wood. The activity of vascular cambium is very high in spring season, and the wood formed in this type of season is called the spring wood. So as you can see the difference between the two types of wood. In case of spring wood, the activity of cambium is very high, whereas in case of autumn wood, the activity of cambium is less. In case of spring wood, due to the high activity of cambium, large number of xylem vessels are produced, whereas in case of autumn wood, due to less cambium activity, fewer xylem elements are formed. The vessels are with wider cavities in case of spring wood. The wood is light in color and is of lower density, whereas in case of autumn wood, the vessels are of narrow cavities, darker in color, and it is of higher density. Now the autumn and spring wood is formed in the form of a ring. A ring of autumn wood and a ring of spring wood is collectively called the annual ring. You can see that these rings actually are formed by the formation of this autumn and the spring wood. It appears as a form of ring, and it gives the idea of a age of a tree. And the study of this age of tree is called dendrochronology. So this term is very important and is one of the most important question in NEET exam, that what is dendrochronology. So basically the study of determination of age of a plant is called dendrochronology. So now I discussed the secondary growth in the stellar region, that how the cambium actually divides. Interfascicular cambium and intrafascicular cambium is formed. Interfascicular cambium is formed, Two types of cells are present: fusiform initial, ray initial, and how by the activity of fusiform initial the secondary xylem and secondary phloem is formed, and how by the activity of ray initial the vascular cambium or the secondary medullary rays are secondary medullary rays or the vascular rays are formed. So this I discussed. So what remaining is that the secondary growth in the extrastellar region. Now secondary growth in the extrastellar region takes place by cork cambium. It is also known as phyllogene or the extrastellar cambium. Cork cambium is formed from hypodermis. Now hypodermis actually differentiates to form cork cambium, which is a type of secondary meristem. Cork cambium also divides perclinally, that is parallel to the surface. It forms cells toward the outside, that is toward epidermis, and some cells toward the cortex. Now these cells form toward the epidermis, their middle lamella is suberized, and due to this the cells become dead. So these cells are known as the cork or phloem. Whereas those cells which are formed toward the inside are differentiated into parenchyma, and these cells are called the secondary cortex or phyllodome. So, phylogene, that is your cork cambium, phylem, that is your cork, which is formed toward the epidermis, and phyllodome, that is your secondary cortex, which is formed toward the toward inner side, are collectively known as the periderm. Now, all the tissues outside of cork cambium, that is cork and tissues which become dead, are called the rhytidome. Now, most of the cells in the phylum are dead, but some living cells are also found. These are called lenticels. Basically, lenticels are made up of scattered group of living cells. These are the living cells, and these are completely scattered. You can see that this is the phylogene, this is the secondary cortex, and these are the cork or the phylem, which is the form by the division of this phylogene. And this is the epidermis, and these are the groups of those living scattered living cells. Now, these scattered living cells are known as the complementary cells, and the scattered living cells actually form the open space, and this open space is called the lenticels, which are formed in the stem, which helps in exchange of gases. Now coming to bark. Now, all the tissue situated outside the vascular cambium is called the bark. Now bark can again be divided into two types, that is the outer bark is called the rhytidome, and the inner bark, that is called is made up of the secondary phloem. So these are the topics I discussed: the secondary growth in the dicot stem. Now the last topic remaining is the secondary growth in case of dicot root. In case of dicot root also some type of secondary growth is seen, and the difference in case of the dicot root, the vascular cambium is completely secondary in origin, as we saw that in case of dicot root, intrafascicular cambium is of primary origin, whereas the interfascicular cambium is of secondary origin. But in case of root, the vascular cambium is completely secondary in origin, and it's originated from a portion of pericycle tissue. The rest of the events are similar as that of the dicot stem and in case of dicot root. So with this I came to the end of this chapter, and also came to the end of this video. I discussed the secondary growth. So I will provide you the notes. Go through the notes, go through your textbook, and in case of any doubt, let me know. So